Yeast, fly, worm, and weed. Part 2. Previously established reputation. This holds for yeast, C. elegans, Drosophila, mouse, and rat. These species have already proven their usefulness as models before the genomics revolution and were adopted by genomicists because so much was known about their genetics and biochemistry. Genome size. At least in the beginning, a relatively small genome was a major advantage for a sequencing project. The genome size of living organisms ranges across 9 orders of magnitude from 10 raised to power 3 base pairs, 0.001 millibase pair in RNA viruses to nearly 10 raised to power minus 10 raised to power 12 base pairs, and in some protist ferns and amphibians, the pufferfish Tachyfugus rubrips was indeed chosen because of its relatively small genome, one-eighth of the human genome. Possibly for genetic manipulation. The possibility of genetic manipulation was an important reason why Arabidopsis, Drosophila, and mouse became such popular genomic models. The ultimate answer about the function of a gene comes from studies in which the genome segment is knocked out, down-regulated, or overexpressed against a genetic background that is the same as that of the wild type. The introduction of constructs in the genome that can report activity of certain genes by means of single molecules is very important. This can only be done if the species is accessible using recombinant DNA techniques. Possibility for genetic manipulation. Foreign DNA can be introduced using transposons, for example, modified P elements that can jump into the DNA of Drosophila or bacteria such as Agrobacterium tumefaciens that can transfer a piece of DNA to a host plant. DNA can also be introduced by physical means, especially in cell cultures using electroporation, microinjection, or bombardment with gold particles. Another popular approach is post-transcriptional gene silencing using RNA interference (MAI), also called inhibitory RNA expression. Medical or agricultural significance. Many bacteria and parasitic protists were chosen because of their pathogenicity to humans. Other bacteria and fungi were taken as genomic models because of their potential to cause plant diseases, phytopathogenicity. The sequencing of rice was motivated by the huge importance of this species as a staple food for the world population. Adam 2000. Some agriculturally important species have great relevance for ecological questions. For example, the bacterium Cynorhizobium meliloti, a symbiont of leguminous plants, is known for its nitrogen-fixing capacities, but it also makes an excellent model system for the analysis of ecological interactions in nutrient cycling. Together with its host Medicago truncatula. Yeast fly, worm and weed part 3. Biotechnological significance. Many bacteria and fungi are important as producers of valuable products, for example, antibiotic medicine, vitamins, soy sauce, cheese, yogurt, and other foods made from milk. Other bacteria are valuable genomic models because of their capacity to degrade environmental pollutants. For example, the marine bacterium Alcanivorax borcumensis is a genomic model because it produces surfactants and is associated with the biodegradation of hydrocarbons in oil spills. Rolling it all, 2004. Evolutionary position. Evolutionary position. Whole genome analysis. Whole genome analysis of organisms that are at crucial or disputed positions in the tree of life can be expected to contribute significantly to our knowledge of evolution. The C-squared CI intestinalis was chosen as a model because it belongs to a group, the Urochordata, with properties similar to the ancestors of vertebrates. The study of this species should provide valuable information about the early evolution of the phylum to which we belong ourselves. The velvet worm Peripatus, traditionally seen as a missing link between the arthropods and annelids, but now classified as a separate phylum in the Panarthropoda lineage. Comparative purposes. Comparative genomics is developing an increasing array of bioinformatics techniques such as synteny analysis, phylogenetic footprinting, and phylogenetic shadowing, by which it is possible to understand aspects of a model genome from other genomes. One of the main reasons for sequencing the chimpanzee's genome was to illuminate the human genome, and a variety of fungi were sequenced to illuminate the genome of Escherichiae, means genome of Escherichiae. Ecological significance. Since the completion of the human genome, ecological arguments have played an increasing role in the selection of species for whole genome sequencing, and we expect them to become more important in the future. Jackson et al. 2002 have formulated arguments for the selection of ecological model species, and we present them in a slightly adapted form.